Hey guys, so we are looking at chapter 15 here today, which is called the human genome. So we're going to tie this in this time with chapter 12 a little bit, and we're going to continue on with what we were looking at towards the end of chapter 12, which was non-Mendelian types of inheritance patterns. So in other words, things that violate Mendel's rules, things that aren't consistent with just the normal dominant and recessive types of relationships. And so with chapter 15, what we're going to look at specifically is going to be human chromosomes, human genes, and how those sometimes can be inherited differently, and also how we can kind of track those through other types of models. So as part of chapter 15, we're going to start here looking at chromosomes again. And in this section especially, you're going to hear a lot of things that should sound like review because they are. There's quite a bit in this section that ties back in with um, chapter 11, actually, even going all the way back to where we started looking at meiosis and different types of chromosomes, that kind of stuff. But certainly with chapter 12, you're going to hear a lot of similarities here, and then we're going to build on some of those concepts presented in chapter 12. So again, make sure you've got the title here. For our objectives for section 15.1, we're going to look at karyotypes, what those are. You've seen a picture of a karyotype before. I'm going to show you a couple more here in this section and look at what they can be used for. We're going to talk about other types of inheritance patterns, namely what we call X-linked or sex-linked inheritance. And then we're going to look at things called pedigrees, which is how we can track family histories of things like disease. Okay. So starting with karyotypes first, this is what you're seeing here on the screen is a picture of a karyotype. This is called a karyotype, I should say. So karyotypes represent all of the chromosomes that we find in a given body cell of any individual organism. And we can make these, primarily we like to make these if a couple who's having a child maybe has questions about the viability of that individual. Like perhaps sometimes, and this is can be the case maybe more frequently than we'd like to consider, but you can have abnormalities with chromosomes. And so what karyotypes look at is the structure of the chromosomes and also the number of those chromosomes. All right. So when we start talking about chromosomes and the 46 that make up human body cells, we can kind of use this word also here to kind of infer what it is that this has to do with genetics. And when we think about a genome is all the genetic information that we store within those 46 chromosomes. Okay, so we have what's been identified as a human genome, and that's actually, I'm going to scoot back here for a second. That's actually what's represented right here is kind of the discovery of the human genome. And as we get into the 90s here, this is called the Human Genome Project. And all these events kind of transpired and happened that allowed us to really have a pretty solid understanding of the genes that we find in our chromosomes. Now, that doesn't mean that we know exactly what each gene does in our chromosomes, but we have to first kind of understand the sequence, right? We have to know, what is identifying as a gene? What is it that we use to kind of distinguish a gene from something that's not a gene? And in doing that, you have to have an understanding of the chromosomes where we store all this information, right? So looking at a karyotype, looking at the chromosomes is kind of like the first step to diving into this concept of understanding the genes that we find within these structures. So again, we can use these pictures to kind of assess whether or not an individual has the correct number of chromosomes. And not that we can do much about it if they don't, but certainly, that would give us some understanding in terms of what our expectations should be and how that individual might be affected. So the way that these are taken, these are imaged from a cell that's been going through mitosis, because again, we're talking about body cells. These are chromosomes that have condensed. And when they take a picture of these, they're going to kind of, using a computer, they're going to model and move them around. They're going to snip them, crop them, and move them to other places where they line them up properly with their homologous pairs. So you can see I have pair number 1, homologous pair number 2, 3, 4, 5. And as we talked about before, we number our chromosomes just really for simplicity's sake. We go from largest to smallest. And then in the very right-hand side here, you can see I have one additional pairing that's being highlighted there, which is what we call our sex chromosomes. So if you remember from previous screencasts and lessons that we've done, our sex chromosomes determine an individual sex. at birth. So if you are XX, then you are born a female. If you are XY, then when you're born, your sex is male. And there's really not much kind of in between that when we talk about, genetic things. You could have scenarios where we have different numbers of chromosomes, but in almost all of those scenarios, that having that condition results in certain death, early premature death. And so there aren't a lot of exceptions to this rule really for humans, that we have to have 46 chromosomes and no more and no less. Okay. But in terms of some of those unique numbers that we could potentially inherit, what we can look for and see in these karyotypes are differences that maybe would be unexpected otherwise. So we could see what we call chromosomal mutations, which is where we have pieces of chromosomes that are either added and are extra on top of something else. We could see duplications, where maybe we have a whole segment of a chromosome that's been copied and kind of stuck on the end. We could see insertions, where we have pieces of chromosomes that don't belong there that have been inserted into something else. And we can also see extra chromosomes or deletion of chromosomes if that's happened between our homologous pairs. Anytime we have extra or too few chromosomes, we use this term aneuploidy. That's how you say that. And aneuploidy just refers to this idea that we have an abnormal number of chromosomes. So for humans, the only two real aneuploidies that are possible would be monosomy, which is where we have one too few. So instead of having 46 chromosomes, we only have 45. This is almost always fatal with the exception of the sex chromosomes. So if we lack, so again, if you remember, XX codes for a female, XY normally codes for a male. There are exceptions where, and this is called Turner syndrome, where a biological female, okay, an individual who's born with the sex of a female is born with only one X chromosome and nothing else. So instead of having 46 chromosomes, she's got 45, and she only has one sex chromosome. For most of those scenarios, individuals with that condition, if that's the only chromosomal abnormality that they have, they tend to survive and they'll do so well, but they can't themselves reproduce. Okay. So most monosomies, if we're lacking one of our 46, are going to be fatal and deadly. The exception would be something like Turner syndrome, where we're lacking something as small as a Y chromosome. Okay. Trisomy is kind of the opposite. This is where we have 3 chromosomes where we would normally have a pair. So Down syndrome is caused by trisomy number 21. And if we look at this karyotype, chromosome 21 is down here. It's really, really small. But if you know anything about Down syndrome, there are some significant effects of having that one extra chromosome, even though it's teeny, teeny, tiny. So if we consider that, the impact of a disorder like that and having an extra chromosome, you could imagine how much more severe it would be to have something like an extra chromosome 4, which is much larger and therefore would likely contain many, many more genes, right? So if we look at this karyotype, this is an example of a karyotype for an individual that would have never survived to birth. Okay, we're lacking part of chromosome 1 here. We have a missing piece of chromosome 3. We have an extra chromosome 4. Extra chromosome 18. Some of these chromosomes just don't look like they were formed or maintained properly either. So this individual would have been a male. You can see there's an X and a Y down here, but again, would never have survived because chromosome number is just kind of all over the place. Okay. So when we think about chromosomes and chromosome numbers, one of the things that we've talked about before is the difference between are 2 different categories of chromosomes. We have autosomes and we have sex chromosomes. Autosomes, if you remember, are those chromosomes that we number. So they're the 1 through 22, and they have genes for non-gender specific things. So again, the traits that we all kind of share in common, like hair color and eye color and height and all that. It doesn't matter if you're male or female, we all have these in common. And so those autosomes are the ones that we number. So in a body cell, that means we have 44 autosomes or 22 pairs. And in that same body cell, we would have a pair or 2 sex chromosomes also present. So that's often defined as the 23rd pair when we consider a human genome. The X and the Y are both important, especially in males, but we know that the Y chromosome itself is especially important in terms of developing male-specific characteristics, okay? The X, on the other hand, here's a picture of our X chromosome. The X, on the other hand, if you can see some of these things that are listed, like we have deficiencies, we have cleft palate, we have hemophilia, which is a bleeding disorder, we have color blindness. These are not things that we typically associate with females, right? This is not a female-specific chromosome. So unlike the Y chromosome, which is very much in tune to sex-specific types of traits, the X chromosome does a lot of other things. So we know there's more than 1,400 genes found on that X chromosome. Of those, we're only labeling, a very small percentage of that here. But when we think about what it must be doing, the X chromosome is super important in that it's providing this information for all organisms, regardless of if they have two X's and are female or an X and a Y and they're male. Okay, so that specifically that X chromosome becomes super important. So when we think about human genetics specifically, all right, we can talk about our traits as being in one of two categories based on where we find them in our chromosomes. They can be autosomal, which means that they're going to code for, again, body specific things, and they're found on chromosomes 1 through 22. Males and females, given that we're all human and we all have 22 pairs of these things, inherit these in the exact same way. So there's an equal probability, regardless of if you're born a male or a female, of expressing these traits. Okay, so it has nothing to do with sex in this case. So I've got some examples of human-specific autosomal types of traits. So we have the ability to roll your tongue, cleft chin, which is this, which is kind of that, like a, for lack of a better word, like a little butt chin, right? It has a little dimple down here and it folds inwards. A widow's peak, which is where your hairline comes to a bit of a point, not maybe as severe as this guy's, but there's kind of a peak there in the middle. Earlobe attachment. So we've got free earlobes like these and attached earlobes where you can see it's just kind of going straight towards that neck area. Dimples are autosomal hitchhiker's thumb, which is the ability to kind of push your thumb back like that when you just put thumbs up. And I don't have that ability. But, interestingly enough, my brother does. And so he inherited that trait. Now, these types of inheritance patterns, these traits that we're looking at, can be dominant, they can be recessive. We're not talking about that right here. All that I'm referring to in regards to these, I'm just saying that these are examples of genes and traits that we find specifically on our on our chromosomes 1 through 22. Okay. There are some, though, like we talked about at the end of chapter 12 in section 12.3. We do have some autosomal traits and genes that don't follow Mendel's rules. So the ones we just saw do have a single dominant and a single recessive allele, and it's one or the other, right? Either you have dimples or you don't. Either you have the hitchhiker's thumb or you don't, right? There are some, though, that we've mentioned before that don't follow those simple types of Mendelian rules, right? It's not just dominant and recessive. And these would be things like blood type in humans. So if you're A blood or type B blood or AB or O, skin color, hair color, texture, height, weight, eye color, lots of other human characteristics. And these were all pretty much covered in section 12.3. So just recognize that there are a couple of different ways that we can inherit these autosomal traits and express their phenotypes. But what they all share in common is the fact that they're on those chromosomes 1 through 22. Okay. In comparison to that, I mentioned that there's another category of traits that we can inherit, and these traits are what we call sex-linked traits. All right, and sex-linked is a term given to any type of gene that's found on the X chromosome specifically. So there are human traits and also some disorders. So where we have things that go wrong that are tied to that X chromosome. And the picture I showed you back here, I can go back really fast, maybe too fast. Right here, these are all disorders that are tied to specific genes and their relative locations on this chromosome. These are not the normal conditions. And obviously some of these have the word syndrome or deficiency, and that would imply that there's something wrong. So we have genetic disorders that we can inherit on our sex chromosomes. Certainly we can also inherit those on our autosomes. But when it's on the sex chromosome, it becomes a little bit more interesting in a way. So again, keeping in mind that females have 2 X's and males only have 1 X, there's a discrepancy here when we start talking about sex-linked traits. Males, because they only have 1 X, are more likely to show and express whatever is on their 1X. So what I mean by that is, if we look at this Punnett square right here, okay, if I'm crossing and looking at the inheritance of sex chromosomes, I know females have 2X's, so they can only give to their offspring this X or this X. Males have an X and a Y, right? So they can only give this X or this Y. And if they donate their Y in the form of a gamete, right, and then that gamete is used to produce offspring, you could see that the male offspring that result are just going to have this one single X, right? Well, it doesn't matter what's on the Y. The Y is not homologous to X. So what's on the X is not the same as what's present on the Y, right? The Y is responsible for doing male-specific traits. The X, like I mentioned before, does a lot of other stuff in addition to that. So this can kind of lead to some problems for individuals that are born male. And the reason for that is because they only have this one X chromosome. So whatever's on that X, it can be dominant, it can be recessive, it can be incomplete dominant, codominant, any type of inheritance pattern you can possibly think of. If it's on the X and it's only on the X, we don't see it on any other autosomes, males will always show whatever that is. It doesn't matter if, what category it fits into, if it's dominant or recessive, it will show regardless because it is the only copy of that trait that that male has. Females, on the other hand, because we have 2X's, we kind of follow the normal expression pattern for things that are on the X because we have 2 copies, just like we have 2 copies of everything else. So what makes males different in this case, again, goes back to this idea that they've only got 1X, and whatever's on that 1X is always going to be expressed, okay? So males are often more likely to express disorders on their X chromosomes, like colorblindness, like hemophilia, like muscular dystrophy, and some of the others that you saw on that previous slide, okay? These 2 images here are pictures of, and if you've ever been to the eye doctor, and like I go for my checkups every year, they usually make you look at cards like this from time to time, and you're supposed to tell them what numbers you can see. And for me, I can see them because I am not colorblind, but inevitably every year I'll pull up a couple pictures like this and we'll take the test, and I'll have students in my class that say, I can't see those at all, right? And they know they can't see them. This is not news to them, but this is at the point when they're also like, oh yeah, by the way, I'm colorblind. And that generally happens, at least in a few cases, every single year. Never have I had a scenario where I have a female student who is colorblind. So again, the idea is that males are kind of disproportionately affected by some of these disorders, given the fact they only have 1X, all right? Okay, so human traits and disorders, like I said, can be sex-linked and found on that X chromosome. We can do Punnett squares that represent these kind of unique inheritance patterns, and we'll talk about that here in a few minutes. I think I'll pause and pull up a Punnett square to do separately up on the board. But when we talk about sex-linked disorders or sex-linked traits, females can also do some pretty unique things with theirs. So females have the ability to turn off an entire chromosome. So females turn off an X chromosome, and when that happens, we say that the turned off one, the one that's no longer going to be shown in the phenotype, is what we call a bar body. So those X's have genes that never get expressed, and so what can end up happening sometimes is we get this unequal kind of expression of traits in unique patterns. The cats that you're looking at in this picture are all called calico cats, except for this one male cat right here, which you can see he doesn't have that kind of cool orange, black, and white pattern of the mom and his 2 siblings here that are females. And the reason for that is calico cats have the ability to, really, I should say, cats of this type, not just calico. Calico is a term that we give to this trait itself. But cats like this have an orange allele and they have a black allele. And if you look at the way we're writing these genotypes, these are X-linked. And so the way that we write genotypes is we kind of tie the allele to the X to represent that it matters what the gender is. So here, this female cat is X big O, which is the orange allele, and X little o, which is a black allele. So wherever this cat turns off X little o, and that becomes the bar body, the cat creates this orange phenotype because the only one that it's going to express is that dominant allele. Now, vice versa, if the cat turns off that X big O allele, The only thing that shows up in terms of fur color is going to be X little o, which creates a black phenotype. And you can see where we have black spots there. Same happens here with mom, right? Where we see black, she's turned off the X big O, which creates an orange phenotype. And same here for this kitten. Males, because they only have one X, males that are part of the same group of cats are either orange or they are black. But male cats can never be calico because males don't have the opportunity to inherit two X's and then turn one off to get that kind of spotted splotchy pattern that we see here. Okay. So for males, it's kind of it's different, and this makes these types of Punnett squares a little bit more complicated, I should say, or maybe a little bit more important to track gender, right? We weren't doing that at all with the other Punnett squares that we've done, but we will do that with these. So let me pause since I'm done with sex linked. I'm going to go up here and do a Punnett square for you, and then we'll come back and finish the rest of the notes, okay? All right, so let's look at sex-linked inheritance patterns in humans, all right? So some of these, again, this can be one of those things that does not follow Mendel's rule, or rules, I should say, but it's going to be something that you're going to be responsible for being able to do in terms of Punnett squares. So when we talk about sex-linked, I often call it X-linked. They sound the same, and they are the same. It helps me to remember they're X-linked because this is something, again, that we find on the X chromosome specifically. So again, what would be helpful here is to represent those genotypes for females and males, and that's where I'm going to start just as a general reminder. XY is male and XX is going to be female. So when we think about X-linked genes, the way that we're always going to represent them is actually attached to the X chromosome. So let's say I have an X-linked trait of colorblindness, because that's one that we tend to have quite often occurring, or at least pretty frequently, and it's not super harmful, so it tends to be one of those that we can talk about, without really kind of making people nervous about something. So color blindness, I know, and we'll talk about this as we get into the next section, but color blindness is an X-linked recessive trait. So if you have the X little a allele, this means that you are colorblind. Okay, but notice it is recessive, right? So if I have X little a, technically I would need 2 copies of that little a allele to actually show color blindness, okay? X big A, on the other hand, would be that you're normal, or we're going to say in this case, no disorder. All right, so no disorder in that case. So if I look at a cross between maybe, let's say I have a pair of parents where I've got a female who is X big A, X little a. And I need 2 X's again because I know she's a female, right? So this is going to represent my female parent, and she is not colorblind either. She has this allele, the normal big A allele, and she's got this one that would normally create colorblindness, but because she's got the big A allele, that's going to overpower the little a, right? Just like we've seen before, this is the dominant allele. If that's present, that's what you actually see. So sometimes females may not know that they're carriers for these things because we have 2 X's and because we don't show what's here, this recessive allele, okay? I'm going to cross her. Let's say that we have a female and we're going to cross her with a male who's also not colorblind, okay? So this is going to be my male, and I know it's my male because, again, I have a Y chromosome here, and that's always going to tell me I've got male, okay? So when we think about crossing this, it looks like it's more complicated, but I want you to keep in mind that it's going to be the exact same thing as what we've been doing this whole time, right? As what we've done before. It's just now my alleles are written like this, right? You have to keep all that kind of together, but don't let that throw you off because it's the same thing we've done, okay? So my gametes from these parents, I can get X big A, right? Or this mom could give an X little a. Dad over here could give an X big A or his Y, right? And depending upon what he gives, that will determine, what our Punnett square and our offspring are going to look like. So I'm going to scoot over here and set up this Punnett square. So let's say I'm going to put mom here across the top, and I'm going to put dad here across the side. And in setting this up, I'm just going to cross them just like I've done before. I'm just going to keep my X's and my A's together, and my X's always need to be written first, okay? Just like when we write a genotype for a male, we write XY and not YX. So here's my Punnett square that I get kind of through that cross and out of that cross. And you can see some unique genotypes. In reality, I've got 4 different genotypes. All right. And so if I want to characterize some of these individuals, I can jot down, what these represent. So this individual here has 2 X's, which tells me she has to be a female. And I also know she's big A, which means she's not colorblind, and she's also not even what we call a carrier. She doesn't even have the allele. She doesn't even have the allele to pass on to offspring anyway. So she's a female, not a carrier, because she doesn't even have the little a allele to pass on. Okay, this one, on the other hand, is a female, but because she's got that little a allele, we're going to call her a carrier. She is not colorblind, but she carries this little a and can pass it on to her offspring if she passes down that X chromosome, right? 50-50 chance of that. This here is a male, and because he only has the big A allele, he is definitely not colorblind. He has no disorder. He's also not a carrier. So he's male, no disorder, and not a carrier, right? And I could have also added here no disorder for this female. And then I've got this other male down here who's the exact opposite, right? I mean, I have another male, but in this case, the only A allele he has is the little a, which we wrote right here represents colorblindness. So X little a means I have a colorblind male. Now the question then is, where did he get that little a allele from, right? Who's responsible for passing that down to him? And if we look back at our parents, we know that the little a allele came from mom, right? So anytime a male inherits an X-linked disorder, it's always going to come from the female parent. It never comes from the male. The only thing that a male passes on to his offspring that are also males is going to be the Y chromosome, okay? For a female to inherit a genetic disorder like this or any type of trait on the X that's recessive, she has to get it from both dad and mom, right? So if dad here had been colorblind, if dad here was actually X little a, then that would mean in this box right here, I would have a female that's X little a, okay? And in this box right here, I would have a female who's actually a carrier. But nothing else changed. Notice this doesn't impact his sons, the male offspring. It only impacts the female offspring because he is what's kind of passing things down to those females, to his daughters, okay? So in this case, you would have a female that's colorblind. But again, that's only when you have a male that's colorblind and a female that's a carrier, or a female that's colorblind and affected in this case, okay? So again, these are set up just like what we've seen before. Again, you're just keeping your alleles, your A's in this case, for the one we set up, with the X's like tied to it, because in this case, gender makes a difference. In all the Punnett squares we've seen up until this point, gender did not matter. It didn't matter which parent was female and male because it didn't play a role in determining the phenotype, right? But here it matters because males we know only have that one X, and as a result of that, they're going to be kind of disproportionately affected by whatever's there. Either, it's recessive and they show it, or it's going to be dominant and they show it. They can never cover up an X-linked trait, regardless of if it's dominant or recessive. Okay. All right, so let's go back to the notes. Okay, so pedigrees, moving on to the next little section of this section, kind of weird, but pedigrees, when we talk about these, this is a different way of monitoring the transmission, the movement of a trait, especially within a specific family line. So these are kind of like a family tree, but what makes them unique is that we're representing individuals as symbols, and those symbols have meanings attached to them. You will see examples of pedigrees on homework, you will see them on your quiz, and you will see them on your test. And I'm going to try to make another portion of the screencast here in a minute that will help kind of go through for you exactly how to read these things. But if you look on this slide, and by the way, it would be good to jot this information down so that you've got like in a way a key of what's represented in these types of symbols and diagrams. So when we draw a pedigree, a couple of general things to keep in mind. Like I said, this is a family tree. The oldest generation, the furthest back in time, is going to be up at the top. Okay, so these are like the original parents. Anytime these parents produce offspring, right, and couple to produce offspring, usually you'd say get married, but we all know that couples don't have to be married to have kids, right? So this couple here, and again, it has to be between a circle and a square in this case, because we're talking about combining genetic information. When these two create offspring, we have a little vertical line here that's going to represent their specific children, and it comes directly off of the horizontal line that was connecting the pair of parents. That vertical line will go down, and anybody that branches from that, those are going to be the children of that original pair. So you can see from here, they have 1, 2, 3 children. Horizontal lines here that are connecting these outermost children are going to again represent who they're interacting with to then create children themselves. These are not children of this original pair. Notice they didn't branch off from the original parent couple. But they did join the family and then therefore contribute DNA to the offspring and their own children, which are represented then here, right? You can continue going down as you get to more recent history. A couple of other things. Circles in these diagrams are always female. Squares are always male. If we have a filled in shape, whether it be circle or square, that's going to represent the fact that that individual has the trait. And when I say the trait, I'm trying to be kind of generic because the trait can be lots of different things. It could be the presence of a disorder. It could be the presence of the hitchhiker's thumb, right? It could be the presence of blue eyes or type O blood or whatever. But whatever it is we're trying to investigate and look at, that's what the trait is, okay? If you have an empty circular square, that means that that individual does not have the trait. So importantly, we're not using genotypes to construct these things. We're actually using phenotypes to make these pedigrees because it's based on what you see. It's based on what's expressed. So sometimes we can come up with the genotypes based on these symbols, but sometimes you may not be exactly sure. It may be like 50-50. It could be this or it could be this, right? So if we look at kind of how these are used, again, we're tracing the expression of a trait. We're going to use phenotypes to infer from those the genotypes as much as we can. And then we can also try and use these to identify if a trait is dominant or recessive. Because oftentimes when you see these, I'm not going to give you a lot of background information about what it is we're looking at. I'm just going to ask you, okay, if someone's affected, who is that someone? How were they affected? Where did they inherit that from? And what type of trait might this be? Could it be that it's dominant, or could it be recessive? Could it be autosomal, or could it be sex-linked? Okay. And so with that, those are really the two major questions that you get asked with pedigrees like this. Now on this one, you can see they're labeling my generations for me, which is really nice. They're also labeling for me what's dominant and recessive. So this is almost like a pedigree with a nice key. And you can see this trait where we're filled in. Those are the individuals that have the trait. And so this is a dominant type of trait. Anybody who's filled in has the big W or the dominant allele. So this is a trait that would be dominant. We also know it would be autosomal because it's not represented as being attached to that X chromosome, right? We could also say it's autosomal though, because I have females that are affected, and actually I've got more females that are affected than the males. And that will not happen if it's something that's sex-linked, okay? And that's just one of those rules we'll talk about with pedigrees here when we go through them up here. In addition to that, if we look at this one in comparison, you can see how this one differs. I mean, obviously we're representing genotypes here, and I've got X's and Y's, which tells me this has to be X-linked. But beyond that, if I look at who's actually affected, only the males actually have it. These circles that are half filled, again, they represent females, but the reason why they're half filled, if you look at the genotype, is because these females are actually considered carriers. And if you've not heard that term before, what that essentially means in genetics, it means you have a trait, in this case, the recessive trait that causes whatever this thing is, the recessive allele, but you don't actually show it. So in other words, you're heterozygous, you got it, you don't show it, but you could pass it down. So we call you a carrier because technically you're carrying the allele and you could pass it on in the next, generation or beyond that. Okay. So I'm going to switch gears again. We're going to do some pedigrees back here. So I will be right back in 1 second. Pause if you want to pause, take a break, and then I'll see you there in a second. All right. All right, so let's look at solving pedigrees, the very last part of this section. And I tried to put a whole bunch of different options up here of things that we can talk through in scenarios where we can look at exactly how to figure out what's going on in some of these. So I left it pretty blank beyond just including the symbols and kind of the family tree part, because I don't want us to make any assumptions about what type of trait this is. I want to go into this like you will have to on your homeworks and quizzes sometimes. I'm not going to give you any background about the trait that we're looking at, but what I want you to tell me is what you can identify about the trait based on this, okay? So first couple things. With a pedigree, we first want to identify what the symbols represent, right? So up here, I'm treating this as if it's kind of like my key for this one. So if you remember, the circles represent females, all right? So any circle on this, I'm going to go ahead and label female. The difference between if it's filled in completely, not filled in at all, or filled in halfway tells us whether or not they have a specific trait, okay? So with pedigree, sometimes we use the word affected, not infected, but affected, like you have it, or unaffected for those that don't have it, okay? So if it's a female that's filled in, that means she's affected. She has whatever the trait is that we're looking at, okay? For a female that's not filled in, a circle that's not filled in, she's unaffected. She doesn't have it. She doesn't express it. She is without whatever that trait might be. And then for a female that's half filled in, we say that she is a carrier, meaning she has the allele, but she doesn't necessarily show it. She can pass it on to her offspring, but she doesn't express it in her own phenotype, all right? A lot of times that means we don't even know that it's present, okay? In contrast to that, the squares represent males. All right, a filled-in male, just like a filled-in female, is going to be the male that's affected by the trait and has that physical expression of the trait. An empty square is going to be a male that's unaffected, similar to the empty circle. And then the half-shaded square is a male that's a carrier. And males that are carriers and that are labeled that way are going to be kind of important because we don't tend to see them that often on these things. And we'll talk about why that is a little bit later. The other thing I want to mention with these keys like this and these symbols, it does not matter if it's filled in on the right or on the left. Like I can flip these and it means the same thing. Okay, there's no rhyme or reason why it would be one half or the other that's filled in, but it's always along that vertical line. Okay. So what I've got labeled here, what I've got set up for us are really three separate pedigrees, okay? So if I divide those up and I kind of separate them so you can see the difference. There's one here on the left, there's one here kind of in the middle, and then I've got my last one here on the very far side, okay? So we're gonna handle them one at a time. So as I was saying with the notes, what we can usually ask with a pedigree is two questions. And I would expect you for any series of pedigrees, by the time we're done with all this, to be able to answer these two questions for me. So the first question is the pedigree autosomal or is it X-linked? So I'm going to put that up here as like our first question. Is it autosomal? Does it represent a trait that's autosomal, or does it represent a trait that's found on the X chromosome and would be called X-linked or sex-linked? Okay. The second question is that trait that's being represented, is it dominant or is it recessive? Okay, we've talked a lot about dominant and recessive through doing Punnett squares, right? When we think about dominant and recessive, the same rules apply here. If you have one copy of a dominant allele, right, you should show that. If you have two copies of a recessive allele, you would show that. In general, where we see these carriers that I mentioned before would only be in a scenario where we actually have recessive alleles. If we see carriers on any pedigree, like we're seeing them here, that always is a kind of like a dead giveaway and an indication that we're dealing with a recessive trait. And the reason for that is we're basically saying with a carrier that they have the trait, but they don't show it, right? And we know that we don't have the ability to do that if we're talking about a dominant allele, right? Dominant alleles always show, even if there's only one copy present. So when we think about carriers, as they're shown, these half filled in squares or circles, right? These are always going to represent for us, this specific scenario where we have a recessive trait, okay? So I'm going to actually start with this one in the middle because that's the one that I just kind of talked about. So when I look at a pedigree, I always do a few things. First of all, I can label generations. And on a test or a quiz, this becomes really important because this is how I communicate to you. It would be as if I'm standing here in front of you at the board and saying, tell me about this person right here, right? And I can't do that very well on a test unless I circle it or highlight it or if I have a coding system. And so when we set up pedigrees, there's a system for numbering and identifying individuals. So what I would want you to do is maybe jot this down. It looks more complicated than it is. It's just circles and squares. But as you jot it down, anything I'm going to put up here, you're going to want to also add, okay? So first thing I want to do is just label these individuals. The way that we do that, as I said in the notes, the oldest generation is always written up at the top. And so my oldest generation gets generation number 1, okay, Roman numeral 1. And so we always do the Roman numerals off to the side. And then within that generation, excuse me, I have individuals 1 and 2, okay? Now, if I wanted to identify on a test for you, I wanted to ask you about this person right here, okay? I would write it like this. I would say, tell me something about individual 1-1, right? How could we characterize individual 1-1? And you would need to know, without any labels on the pedigree itself, without me giving you these Roman numerals and such, you would need to know that it's this individual. Okay, so when we number these, they go top to bottom, and then they go left to right, all right? So 1 and 2, this is generation number 2, all right? And I have individuals 1, 2, 3, 4, and 5, okay? So we're restarting our numbering with each generation, okay? The next generation down is generation 3, so Roman numeral 3, individuals 1, 2, 3, 4, and 5. In my fourth generation, so far I only have 1 individual, but I can still label that. That's Roman numeral 4. This is person number 1. Okay, so again, if I just pick another one, let's say I want to know about this person right here. I would say, tell me about the phenotype or the genotype of individual 3-3. Okay, so that's kind of the notation for that. And you would maybe tell me, well, this is a female who is unaffected, right? And it may be to the point where I even ask you what the genotype might be, and we'll talk about how you get there, okay? But when we think about these individual symbols that we're representing here, the way that we can use these to try and answer the 2 questions that I put up here at the top is in considering a couple of different factors. So first of all, in addressing question number 2, where we said, is it dominant or recessive? When we think about that, right, remember going back to what we did earlier in chapter 12, if something's recessive, you have to have 2 copies of that allele to show that phenotype, which means, again, I can have carriers that have just 1 copy of that allele. So that means my answer here, if I think about number 2, I want to answer these 2 questions, I can say that I know that this is a recessive trait. And the way that I know that, and I can even go through and prove it, right? I could even write out genotypes and say, well, maybe this person's this, right? Because they're carriers and I know they have to have it, but they don't show it. But I can say I know it's recessive, and my reason for that is because there are carriers shown. In addition to that, another piece of evidence that we could say for recessive is that it's not all that commonly seen in this family tree. In other words, out of all of these individuals that were being shown, all the circles and squares in this pedigree, only one of them actually has and is affected by whatever this trait is. Everybody else is either unaffected or they're carriers who may or may not pass it down to their offspring. So it's not like it's that common, and that's what we kind of tend to see with recessive traits. Okay, so I'm also going to add an additional explanation here, which is that the trait affects less than 50% of the population. And that's going to be kind of a rule of thumb that I'm going to ask you to keep in mind as we go through this. Less than 50%. And I'm not going to say population, 50% of the family. That would be a little bit more accurate, okay? So because of these 2 things, again, we're going to say this is recessive, all right? And that's kind of the thinking that you need to go through when you see these. On your homeworks, I'm going to ask you to give me an explanation, okay? On your quizzes and tests, you're probably not going to have to give an explanation, but you may see multiple choice type questions that ask you about, why is this recessive? What is true of a recessive type of trait on a pedigree? So it's important to know the explanation pieces, okay? Second question appears autosomal or X-linked. And as you saw from earlier on, if it's X-linked, it tends to be seen more often in males than in females. Autosomal is shared equally. So that's like, your hair color, eye color, skin color, all those things that impact all of us regardless of the sex that we are born with, right? So when we think about sex-linked, we should be seeing in a pedigree a scenario, if it's truly sex-linked, where males are affected, but females tend to not be affected. All right, and I see in this case, I've got a male that's affected. I don't have any females that are actually affected, do I? I've got just one male and that's it. And if I think about the way that this is set up, well, it sure seems like it could be sex-linked, right? Because we know that if a male passes down his X chromosome, he has a daughter. If a male passes down his X chromosome, he's going to have daughters all the time. If he passes down his Y, he always gets a son. But if this male is passing down his X, well, we know he's affected, right? He would have, if I erase these to make it a little less clear, or a little bit more clear, hopefully, he would have to be something like this, because we're saying it's recessive, so it's got to be little a. If we're going to go with this assumption that it's X-linked, then we could write his genotype as X little ay. All right, which means the only X that he's got to pass down is X with a little a. And so the females down here, given that they don't actually have the trait, but they're carrying it, would have inherited that X little a straight from dad. And it would make sense that would kind of work. Dad here would have gotten it from his mother back here, who could have passed it on to him as she was also a carrier, X big A, X little a, right? This female here is unaffected, and we're also going to make the assumption she's not a carrier because she's not being shown that way. So as a result, she was likely homozygous, X big A and X big A, and passed down the normal allele to her daughters. So because of a couple of reasons, because we see males affected and not females, because we see female carriers and not male carriers, and because it's pretty infrequent, this is probably going to be sex-linked and recessive. So my answer for the first question up at the top, I'm going to say it's X-linked. All right, and the reason for that, again, is going to be only males are affected. And females, again, are the carriers, not males. Okay. So that's kind of how you would work through and walk through this problem here. Okay, so this is what we would call an X-linked recessive type of trait. All right, I'm going to scoot this direction. And let's look at this pedigree right here. So again, jot it down or something similar if you want to go back and kind of, use this later to review. This is a scenario, though, where you can see I have 1, 2, 3, 4, 5, 6, 7 individuals that are actually affected, and I only have 1, 2, 3 that are not. So if we consider the second question on my list here, dominant or recessive, if we look at that one first, again, like we did on the last slide, or on the last pedigree, not slide, but if I consider dominant and recessive, first of all, I don't have any carriers, right? So that doesn't prove or disprove this recessive, but we know that in general, if we're shown carriers, it has to be recessive. So I don't have those in this case. I can't really apply that. We also see here that what we wrote for our explanation for this one, the trait affects less than 50% of a family. That's not true in this case, right? This one's pretty frequent. I have a lot of individuals that actually have and are affected by the trait, much more than 50%. And so for that reason, and because we're seeing it in every generation, and actually this should be flip-flopped. That was my mistake. Because we're seeing it in every generation, what we see happening in this case is this dominant allele being passed down and down and down. So this is an example of a pedigree that we would see for a dominant trait. Okay, and the reasons for that, our explanations for that, and I'm disappearing off the screen here, our explanations for that would be every generation. So again, generations being these family groups, so Roman numeral 1, 2, and 3, is affected. So we could say every generation is affected. That's one thing. We could also say no carriers. And then we could also add kind of the opposite of what we put right here. Trait affects more than 50% of the population, so greater than 50% of the family. Okay, so for those three reasons this time, we can say, hey, this appears to be a dominant type of trait. Now, if I consider the other question, autosomal versus recessive, remember, if something is X-linked, autosomal versus X-linked, if something is X-linked, it's going to be more often seen in males than in females. So in a pedigree, we're going to see a lot more squares filled in and perhaps no circles filled in. If I count how many of each I have right here, I've got 1, 2, 3 squares that are filled in, that are affected. And I've got 1, 2, 3, 4 circles, 4 females that are affected, which tells me I've got more females than males. If it was truly sex-linked, chances are it would not be like this. I would have a lot more males, to the point of having maybe only males that are affected, okay? So as a general rule of thumb, if you see females that are affected at all, you tend to go with it's got to be autosomal, because X-linked traits appearing in females like this are going to be pretty rare, okay? So we'll say this one is autosomal. In this case, it's on chromosomes number 1 through 22. And the reason for that is going to be both males and females are affected. Okay, so both males and females are affected in this case. And really, it's about half and half. All right, so this one is, again, autosomal dominant. And you can see, hopefully, how different that is from this one. Okay, last one I wanted to mention and bring up, and let me just scoot this guy over a little bit, is this last pedigree right here. So this is a much smaller one maybe than the others. Again, the labeling would all be the same. So generation 1, 2, and 3, 1, 2, 1, 2, 3, 1, 2, right? I could still go through and number all that. Again, jot this one down if you haven't yet. So here I have a male that has the trait, whatever that trait is. And then I have 2 offspring that they create, this parent couple, that are unaffected, right? They're not filled in. They don't have it. But somehow it reappears down here. And this is kind of an interesting thing that we tend to see often in pedigrees, especially in pedigrees that represent recessive traits, where it appears that you've got a trait in one generation, and that trait is essentially skipping a generation. Like it was there, it disappeared, and then it came back, which is equivalent really to what Mendel saw with his peas, right? He would see like a recessive phenotype, he would cross the recessive and dominant, the recessive would disappear, and then when he self-pollinated those, or when those were allowed to self-pollinate and then cross, he ended up seeing the recessive reappearing. So what that tells us here is that there was actually something present in this generation. It's just not being represented. Now, I could have labeled it and included like a carrier here, right? I could have put that there and say, hey, I know this person was a carrier, and in that case, the male probably was too. However, I'm trying to make a point here, and that point is to say that we don't have to show a carrier on a pedigree. So while this is great, the fact that I have carriers shown here, don't ever count on being shown carriers on a pedigree, because more often than not, they will not show those to you. And the reason for that is if you think back to what we talked about in the notes, carriers are not identified by phenotypes, and pedigrees are representing what we see, the actual phenotype. So if we're using phenotypes and we can't tell who's a carrier and who's not, it's more likely that we're just not going to fill these guys in. Now, we could assume, based on what we're seeing, that they had to have been carriers, right? And we can go back later and do that. We're not going to. But again, that's one of the kind of look for items, one of the rules of thumb that you can watch out for to help you distinguish between autosomal, X-linked, dominant, recessive. Okay. So given the fact that we're skipping a generation, this is going to be a recessive type of trait. So I'm going to go ahead and write that one down. So recessive. And the reason, again, for that is because the trait is skipping a generation. Trait skips, and I'm going to be real specific here because why not? Generation 2 in the pedigree. And we're, it's this pairing right here, especially that we're looking at. Okay. I could also add that I know it's recessive because it's in less than 50% of the family. So I'm going to add trait affects less than 50% of family. All right, and then in terms of the first question, autosomal or X-linked, I can try and infer that from this one too. So again, you want to ask yourself for autosomal, who's affected in this case? And I guess I just wrote down the answer. That was silly, but you want to ask yourself who's affected in this case. And I can see right here, I have 2 females that have it. I have 1 male that has it. And remember, if it's going to be X-linked, you want to have a scenario like this, where only males are affected and not the females. In this case, my females are outnumbering my males again, just like we saw over here in this one. So because I have more females than males affected, that tells me that this has to be autosomal. So more females affected than males. All right, and then from that, what I can do is I can actually say, okay, if I know this is autosomal recessive, and I can do this for all of them, by the way. I did a little bit right here, but if I know that this is autosomal and recessive, then I can go in and figure out genotypes for almost all of these, right? So if we think about what that means, autosomal are those traits that don't include the X with them, right? Like I could just use A's to represent this to be simple. So if this trait, and I'm just going to do this one as an example because it's a little bit smaller, but if this trait is autosomal and recessive, well, I can say, hey, my alleles are going to be big A and little a, right? Recessive would mean that we have it, right? So affected would be the little a, meaning we have the trait, okay? Autosomal then means, again, we're just including the A's here, but the dominant in this case would be the unaffected or the normal condition, whatever the trait is not, okay? So in this case, I could go ahead and say, all right, I know that whoever's affected has to have 2 recessive alleles. So this female here has to be little a, this one here has to be little a, this one here has to be little a. And then I can kind of work my way back to try and figure out the ones that I've left out. So if these 2 offspring have 2 little a's each, they had to inherit those from mom and dad, right? So that means mom, given that she's not affected, had to be big A, little a. And dad had to be big A, little a, because otherwise you couldn't get 2 little a's if someone was homozygous dominant in big A, big A. So they had to inherit these from their parents, which makes them actually both carriers. This one also, this one has a big A and a little a. And in this case, that means that this parent had to have at least 1 big A, right? We also know that this individual here has to have at least 1 big A. But beyond that, we don't know what this individual could have been. We know that this person right here had to get at least 1 little a from this dad. So that individual has to be heterozygous because all that dad could pass down is a little a or a little a. But this mom up here could have been big A, and passed on one of those 2, or she could have actually been big A, little a. And so with that, we can sometimes, go through and identify these genotypes, and you will be asked to do that as well. But again, the key would be you've got to figure out this stuff down here first, be able to answer those 2 questions, to then go back and assign some actual genotypes, okay? All right, so as you can see, pedigrees are not the simplest thing in the world, so we're going to practice, and if you have questions, of course, let me know, all right? Bye, I'll see y'all later.